also my pleasure to introduce Dr. John B. Harley. Dr. Harley is the professor of pediatrics and medicine and director of the Center for Autoimmune Genomics and Etiology, director of the Division of Rheumatology at the Cincinnati Children's Hospital Medical Center and the University of Cincinnati. And he has been a very important figure in moving forward the genetics of lupus, very important figure in our field. So I welcome you.
Thank you, Marta. Thank you so much for inviting me to speak and for bringing us to this wonderful city to have celebrate our progress in our battle to understand the basis of lupus. And so I'm going to talk today about a particular genetic polymorphism. Dr. Kimberly gave us the wonderful overview, the 20,000-foot view of what's happening. This is going to be a presentation in which we get down into the weeds and try to understand the basis of where it comes from. I don't need to tell this audience anything about this slide. The patients with lupus kind of have a smorgasbord of different manifestations, all of which are unified by having autoantibodies. Anti-nuclear and other autoantibodies that these patients share give them risk for these disease manifestations in a way that we don't yet quite understand, but we're placing pretty good dents into it. So here you are, composed of 3.3 billion bases of DNA, divided up into these chromosomes, with the address of all these genes makes a huge difference, and how they work makes us who we are. There's been 3 billion years, we think, of mistakes and errors made to get us to this point. And so to imagine that we're going to understand this in a generation or two of intense scientific study is foolhardy. And to think that this is extraordinarily complicated is how humbling it should be to bring us to this point. Lupus is a wonderful genetic disorder. It has high twin concordance. It varies in different ancestries, with African ancestry being about 3.5-fold and Amerindian being 7-fold higher than Europeans. This lambda S is the risk of a sibling of the same sex having the disease divided by the population, and is a measure of the concentration of the genetic contribution in that nuclear family. And so Dr. Kimberly showed this slide before. I think we are on the verge of having nearly 50 genes that satisfy the multiple testing problem that you face when you tackle that whole genome that I showed you before. And I think before the end of the year, given the studies that I know of that are underway, there will be on the order of 100 of these genes. So lupus can be extraordinarily complicated. This is a preliminary shot of some of the data that's coming from the African-American Genome-Wide Association study. And I had planned to talk about that here back when I was originally invited. But the problem was that we ended up with about 7,000 controls and about 3,000 patients. And we had used 4 or 5 different platforms to do the genetic typing, and it crashed our computers every time we tried to do the imputation. And so that took 8 months, and we finally have the data this past week. So maybe in 3 years I'll be able to talk about that. And instead, I'm going to concentrate on a particular gene. But first I'd like to mention where is it that these genes operate? Bob showed you that graph in which the genes are operating in a variety of different cell types. But I'd like to bring you home back to the B cell. You can see that that first panel, shows the yellow line going above the significance mark that Ray Chowdhury and his colleagues established. This is looking at the variation of genetic expression in the 240 cell types that were available in the mouse and saying, okay, what cell type is likely to have these genes operate? And in lupus, it looks like it's the B cell. In Crohn's disease, it's the dendritic cell, the T4 memory cell, and the NK cell. And in rheumatoid arthritis, it's the T4 memory cell, along with the NK cell. And so, just getting the gene is just the scratching the surface. Where does it work? How does it work? Where does disease risk come from? And we can take half the genes we know about and put them in the NK kappa B and the IRF5 pathway, working through those TLR receptors, B cell receptors, and other molecules circulating that Dr. Kimberly talked about a few moments ago. So this is obviously a really important axis. And we are impressed by this consistency of how frequently the interferon regulatory factor 5 variants are found to be related to lupus. This is one example. And these are all the examples that we had when we put this slide together. 8 different studies, all showing significance. This is 100% of the time that lupus patients are evaluated for association with IRF5. So far, it's been found. And so this also looks like it's the center of the other genes, so it attracted our attention to study this in great detail. This is a transcription factor. It mediates inflammatory signaling, type 1 interferons. It works through the MyD88 and toll-like receptor pathways. It's really important in Mycobacteria tuberculosis and other infectious causes, and it's expressed in B cells, dendritic cells, monocytes, and macrophages at very high levels. It works through TLR7. It seems to be responsible for the homing of dendritic cells. It has a histone acetyltransferates, acetylates, IRF5 converting a silencing complex into an activating complex. It looks like when this gene is disrupted, that metastases and growth of breast cancer cells proceeds with less inhibition. And IRF5 inhibition, in the same sort of thinking, facilitates cell division. So it has multiple roles in the cell and plays an important role in inflammatory responses. And clearly the genotype makes a difference. This is a study by Heidel and Abraham from last year showing the genotypic responses in NOD2 and TLR2 from the homozygote, heterozygote, and the alternative homozygote, showing the increase in TNF-alpha production based upon the genotype of IRF5. And here is a study now in monocytes looking from Barnes' lab, showing the activation and constitutively activated IRF5 in these particular cell types. So we said, okay, how would you get at where this is coming from? How does it work? The typical approach that geneticists are taking these days is to say, okay, let's find a marker, and then we'll find something functional that's nearby that marker, show that it's allelically different with the risk versus the non-risk, and then we'll attribute candidate possibilities that it's responsible for disease risk. And so, being someone who respects the complexity of life, I think that I have the suspicion that that is going to lead us astray. That assumption that you find something that's allegorically different is not enough. That linkage disequilibrium is not strong enough. That we need to do better than that. So on the basis of that intuition, we said, okay, let's take this locus and do everything. Find all the variation. So we use genotyping, sequencing, PCR genotyping, and imputation to collect the data for 7,560 markers in this region. And this graph shows you the output of that. And then we said, okay, we need a really large collection. So we have 8,395 cases and 7,367 controls. And what you see on this slide is what the distribution of association looks like, with now probabilities that there's association nearly at 10 to the minus 50th. Huge effects. And this has a odds ratio of between 1.6 and 1.8, making it one of the strongest in the of the lupus genes we know about. We say, okay, if we have everything, then there's no theoretical additional marker that's outside of our set. We must have the causal polymorphism, the one or ones responsible must be part of this set if we genotyped everything. And so there's no theoretical third party here that we can blame for disease association. And so now we can mathematically model understanding that boundary condition and how this works. And so if we take the top variant one there and say, okay, we're going to condition variation on what that variant contributes and subtract out how it overlaps with all these others and see what's left. And when we do that, we have this piece right here that you can see that still leaves, 10 to the 24th as an effect. And then we pick a variant out from here, maybe the best one, and say, we'll do the same thing with that one. And then look what it does. And when we do that, it leaves an effect in the front. As a consequence, when we put both of them together, we can abolish completely the association with IRF5 with lupus. It leads us, with a lot of other mathematics that I'm not going to go into at the moment, with the conclusion that we have two effects, one in the promoter and one that's a haplotype that stretches from IRF5 and through the next gene called TNPO3, which is a nuclear transporter, and an interesting gene in its own right. And so in that group, we have to have the effect. And so let me talk a little bit about what the other ancestries do. In the top panel here, I'm showing you the two effects with the linkage disequilibrium in colors with the European Americans, and showing that the haplotype is not related, is not in linkage disequilibrium with the effect in the promoter. Meanwhile, in Asians, there's only the promoter effect, as you can see in the bottom panel. When we compare the African Americans to the European Americans, you can see a major effect in the promoter right here. And then the circle is around the haplotype. It turns out that haplotype does not exist in African ancestry. And the existence of this in African Americans is the result of population admixture. These African Americans are 15.8%, something like that, European. And this haplotype is represented by that. And so these are not, that particular haplotype is not polymorphic in either Asian or African ancestry. It means that the component in the promoter is human all the way through all the ancestries that we know, and the haplotype from IRF5 through TPNO3 is only a European effect. Very interesting. And so we start out with something that looks like this, and we end up with two effects, one in the promoter and one in the one with this haplotype. When we take into consideration what happens to the promoter, we can reduce what's going on. With the haplotype, we condition on the haplotype and look at the promoter, we're left with 3 variants. And these variants have a 10 to the 12th difference in probability. Our position is that unless we've made some horrible mistake here, or the imputation is misleading us, or something else is happening, or we don't have the complete variation, that one or more of these 3 markers is responsible for disease risk to lupus in the promoter region across human beings. And so now, instead of studying 7,530 different variants or anything in linkage disequilibrium, our scientific and experimental attention is focused on these 3 variants. And so this just shows our process of getting down to a 22-member haplotype and 3-member variants to divide this up, starting with what I showed you at the beginning. If we do a subphenotypic analysis, we can show you, as Dr. Kimberly talked about already, that anti-Rho, for example, is associated with the promoter variation at this locus. Now, what does this do for the candidate genes that have already been discussed? There are 4 very interesting variants in IRF5. There's a splice variant that changes how splicing happens. There is this CGGGGG that is either present in 3 or 4 repeats. That's an SPI1 binding site near the promoter. There is a indel that alters RNA degradation, with one form having accelerated degradation relative to the other. And there's an exon 6 indel that alters DNA binding. And there are 13 papers published about how these things work and how they are allelically different. If our analysis is correct, then we can exclude this entire literature as being responsible for being causal at the IRF5 locus for being related to why IRF5 contributes to lupus. And so this is the reason why I'm having trouble getting this stuff published. Because it is now trying to change the paradigm about how we think about this. I'd like to mention that IRF5 is really important in immunology in general. And when we actually discover what it is that's going on in lupus, it'll have implications across many fields. This is a figure from Dudley, 2003, that Franco Marancola gave me. And it shows metastatic melanoma after exogenous expansion of T cells and reinfusion, with about a few percent of the patients having a response, and a dramatic response. Here are examples of metastatic melanoma, almost universally fatal, melting away. These patients have the lupus risk allele of the 70 genes that we tried to study in them. The lupus risk allele predicts and increases their chances of having a response to this immunotherapy. And there are multiple other diseases beyond lupus: ulcerative colitis, multiple sclerosis, acute coronary syndrome, Sjogren's syndrome, a response to multiple sclerosis to interferon therapy, rheumatoid arthritis, primary biliary sclerosis, and systemic sclerosis all have associations with IRF5. Given that some of our collaborators around the country with data sets have been very generous to us, we've been able to look at systemic sclerosis and Sjogren's syndrome. Sjogren's syndrome and systemic sclerosis imitate lupus exactly. An interesting difference is primary biliary cirrhosis. Primary biliary cirrhosis is associated with IRF5 in Europeans, but not in Asians. And that would fit with the idea and our conclusion that in primary biliary cirrhosis, the association exists with the haplotype that's present only in Europeans and not with the promoter. And so this kind of approach, I think, is going to have implications for autoimmune diseases and inflammatory diseases and inflammatory therapies that go far beyond our work in just lupus. And then to close, I'm going to mention my science most wanted is this guy, Matt Weyrich, who we just recruited to our faculty. Matt has been studying the binding properties of 160,000 transcription factors in all of biology with Tim Hughes in Toronto doing the informatics. And so his recent Nature Biotechnology paper presents how they're reconfiguring how these motifs and the binding works for these transcription factors. And so using his new approach, we have been able to identify ZBTB3 as a binding factor that is allelically different in the risk versus non-risk, and that, of course, will be the focus of our work to come. And so with that, I'd like to thank my group, particularly Leah Kadian, who is standing right there in the front, for running this project and for helping us get all this data together and reach these conclusions. And thank you for your attention.
So we have time for several questions.
Yeah, I'm sure that this talk was so uncontroversial that there'll be no problems.
Okay, I have a question. Okay. So do you think you can really exclude the other variants if they're not actually part of the haplotype that has to do with the promoter region? Because you have only condition on either a SNP in the close to the large haplotype there, or there's SNPs that are within the promoter.
Well, this talk is meant to be a summary.
I know.
And so we spent months studying every combination that we thought might be relevant. And so there's at least a 10 to the 6th difference between the haplotype. We can either do the single SNP or the haplotype, and we get the same answer. The haplotype, however, is about 10 to the 15th more powerfully associated than any individual SNP, but it doesn't make any difference on the conditional analysis, if that makes sense. And then either the SNP or the haplotype in the TPN03 region or the individual SNPs in the promoter, we don't have any evidence that there's any haplotype operating in the promoter. And we have the region that contains the causal polymorphisms reduced to about 3 kb at the present time using this population genetic approach. And so all the other variation can be accounted for with completely eliminating the association with any other marker at this locus. I don't know what else you'd want to establish. If you do it the other way around, these markers have a residual that's still significant. So these markers have the potential to explain everything else that we see. The other markers do not in the reverse way. There's still association left after their effect is taken into account.
But wouldn't you think that also within the gene and what is expressed eventually as a protein, you will actually have effects from the other markers that are anyhow functional and that could be influencing then the haplotype that you're actually seeing or the markers that you're actually seeing?
I think you're raising a really interesting question. If our approach here is correct, then it raises the new question of, these functional variants have consequences. Why aren't they disease important? Why don't they have consequences for disease risk? And so if our analysis is right, then we need an explanation for why they're not relevant for disease risk. They have consequences. They certainly change how the expression happens, and they look like they change expression levels. And it looks like IRF5 is disease risk because of the level of expression, which your data shows. Then this presents a contradiction that we don't have an answer for yet. Either there's something wrong somewhere, and I wouldn't be the first scientist to have something go wrong, but either there's something wrong or we're not smart enough to think about how to put it all together into one picture.
You have a question?
Thank you. Eric Moran from Melbourne. Just speaking as a rheumatologist and cell biologist who is not a geneticist, I noticed that in your presentation you used the word causal, and the previous speaker seemed to use the phrase associated with, which seems to me to be a very significant difference. Is this a difference in conviction, or is this a difference in biology that is real? The difference between these two ways of expressing what appears to me to be the same thing.
Well, now you're going to talk about philosophy of science. And we could,
Sure.
what we need is a glass of wine and a couple of cigars, and the whole room can participate. But I think what we really like to know is what is actually causal. Well, all that we can do is establish what's associated. So if you are Kuhnian in your approach, then if you can't disprove it, you eventually accept it. If you use consilience of inductions, meaning you arrive at the same conclusion by doing a whole variety of different experimental approaches, then you reach the point where you have belief. Now, belief is in some ways not science, but you reach the conclusion that it must be true because you don't have any other way to explain the data. And so I think those of us trying to solve disease are trying to find cause. Now, we may not be right. We could easily be wrong. The associations look like they're solid, and every time we try to reproduce them, we get the same answer. So, I'm less risk averse that I'll make a mistake than Bob is,
Okay.
perhaps. Is that probably fair? Okay, I think he agrees with that.
Thank you.
Yes, I have actually another question that I would like. It touches on what Bob mentioned in his talk. I like that very much. And also your talk. If you would say that you have all the genes for lupus, do you think that you would be able to classify or reclassify the disease based only on these risk alleles?
No. Simple answer is no. These risk alleles have the effect size is too modest. There's so much else happening. One of the things that Bob and Marta and I are talking about doing next is to try to understand the epigenetics that happens here. That's a whole new level of interesting variation that can have an enormous impact on how these genes are expressed and what happens in the outcome. And so things are so much more complicated, I don't think the genes themselves will be that useful. The audience is full of rheumatologists. And most of the time, we don't need B27 to diagnose ankylosing spondylitis. And so, you use it as kind of a backup. You get it done, but, you trust your clinical judgment about what the phenotype is. And so these are an order of magnitude less impressive in terms of their size. And they're all additive. And so they're not going to be useful in that kind of way at this point, I don't think.
Can I ask a quick question?
A quick question, please.
Yes. So I'm not a rheumatologist, I'm an internal medicine physician, and I'm not a geneticist, but my question is very basic. When we hear about the odds ratio of the different variants, HLA and complement receptors come up first, and with much higher odds ratio than all the other variants you speak about. Mm-hmm. So I can't really understand all that you explained, but I'm sure I will one day. But my question really is why don't we concentrate on those? Do we actually know all about HLA already? To me, like for diabetes, this explains how antigens are presented. It may explain specificity. It may really explain susceptibility to disease. So could you put it in the framework of
So the only disease...
Immunology.
Yeah, okay.
What do we know about immunology?
So the only one that looks to me like it's worked out well is celiac disease, where the antigen is known, the HLA is known, and how the mechanism operates.
And it's not an autoimmune disease.
Well, yes. Okay. It's close, but not. And in these disorders, we don't have a clue. And in type 1 diabetes, 75% of the heritability for type 1 diabetes is wrapped up in the HLA, and yet there must be 1,000 years of investigator time spent on the last, 2% of the genetic risk. And we still don't understand what the HLA is about. And so the HLA is very complicated. It's huge, and there are multiple genes involved. Separating that out is going to be a lot more difficult than what I've shown you today with IRF5. Here we only have 2 effects. We're able to separate it. We can take a sample size that we're able to put together and pull things apart. HLA is so complicated that it's going to take, I think, the next generation of scientists to figure that out. And what we're missing for HLA is a good idea, a really good idea about how it works, why it's associated. I hope you have one, because none of my colleagues that I know have a good idea about why it's there.